Okay, so just to recap of what we went over last week, Austin, did you watch the lecture that I posted? Okay, so we covered screening and we covered mini prepping mainly. Screening, just to remind you, is the picking of colonies off plates and regrowing the said colony by itself, just because that said colony has all the exact same DNA. And we went over the protocol for that and also the inoculation protocol used after you screen for the colonies for inoculating them into broth cultures. Mini prepping is one of the most complicated things we have in terms of protocols, but in a gist, it's the extraction and then purification of DNA, plasmid DNA from bacteria, E. coli. So how this works is you invest a little bit of DNA into the protocol, say 1 to 2 microliters of DNA. No, microliters. It's more like, what, 500 nanograms of DNA, something like that. It's dependent on your concentrations of DNA. But your goal is to put a little bit in and then get 5 to 10 times the amount of DNA out at a much higher concentration. So you always have some sort of, if it goes well, some sort of net product that you can use the DNA for other things. If you didn't watch the video, I linked it again. This is a hands-on demonstration of the protocol you can watch in order to understand the mini prepping protocol better. Okay, so why do we want to cut parts, right? We've already gone over using plasmids and transferring them between different bacterial colonies and then screening for them, but why do we want to cut parts? Because just doing that's not enough. Well, we want to cut parts because we can add and subtract genes in order to change the function of the plasmid of interest. So this is a diagram of a BioBrick part found in the iGEM registry. It's the standard backbone, your origin, your antibiotic resistance. Here's the part itself, and then it has restriction enzyme sites on both sides of it. I'll be going on to those more in the next few slides, but say, for example, your goal is to have your promoter in front of your part because this part by itself is not going to do anything. It's just a coding sequence for a protein, and without a promoter to actually enact it, it's not going to translate it properly. So our goal here is to get this part and put it behind or downstream from the promoter in order for the promoter to activate it. So how do we do that? We're going to have to use digestions. And so restriction digestions is using restriction enzymes to cut plasmid DNA and thereby remove your part or gene of interest. iGEM uses specifically 4 different types of restriction enzymes, those being EcoR1, SPE1, XPA1, and PST1. There are more enzymes that they use overall. We have a few more enzymes, but these are the ones that they predominantly focus on because they're the easiest to work with. So restriction enzymes are extremely specific when it comes to where they cut. So they look for specific nucleotide sequences within the plasmid DNA. They read it and then they attach and cut it there. And there's different types of cutting. We'll go into that later. But they only cut where they're supposed to cut. They should only cut where they're supposed to cut. If you let them go a little bit too long, they're gonna, cut willy-nilly all over the place. And so the goal there is to have them cut what you want and then denature them so they stop cutting. So the goal here is to, in your backbone, these are these sites that they find. Your goal is to cut them there. So yeah, these are the restriction enzyme sites. The E stands for EcoR1, likewise for the other ones. And the iGEM backbones have these sites built into them for easy cutting, so we can just move parts and attach parts easily compared to just going into nature and having to add a bunch of nucleotide sequences to the end of a part in order to make it capable of being ligated together, so glued together. So we can cut it out now. We can cut out this part that we want using these enzyme sites, but how do we determine what enzymes to use? Because there's 4 different enzymes here. For this side, you could pick either of these, and for this side, you could pick either of these. So we need a way of determining which ones to pick so we can get the parts properly functioning when you attach them together. And how we do this is the ends, right? So the ends, there's 2/3 different types, at least for iGEM. Blunt ends, which are cut like straight, so they don't have any overhangs. And then there's also sticky ends, which are the product of space cutting. So right here, you can see it cuts here, then it cuts 4 nucleotides over. And so when you're thinking of this, think of puzzle pieces. When you have 2 puzzle pieces that don't have any holes to snap them together, you're just putting 2 pieces together and it won't stick. But if you have a piece that has a hole and a piece that has the little knob sticking out, and you put them together, they'll stick together and you can hold it. And so this is the basic concept of what's happening here. Is when you cut it, you're going to have this overhang right here, and then this overhang on the other side, this part. And you can use these overhangs to match parts up. So if one part is cut with EcoR1 and another part is cut with EcoR1, you can attach them together using ligase, which is an enzyme that catalyzes the bonding of both of those. So now we have a way of cutting a part and cutting another part and putting it together. However, we don't really have a way of getting them in sequence properly. So we want our promoter to be in front, upstream from our coding sequence. And we do this, okay, I guess I'll go into this first. So you have to have some foresight in order to do this, right? You have to pick the right enzymes and put them in the right parts in order to get them to have one piece to have an attachment and the other piece to have the alternative attachment so they fit together. Luckily, iGEM has a thing for that, but also you can use 2 restriction enzymes in the same tube at the same time. So back here, say there's this part, we don't have to cut it one by one. We don't just have to have this plasmid in there with XPA1. We can have it in there with XPA1 and PST1. So you can cut it on both ends at the exact same time, with one end having the XPA1 overhang and the other end having the PST1 overhang. And that's called double digestion. So here's the foresight. Luckily, iGEM has this procedure detailed for us. For your mini projects, you're going to be using 3A assembly, which I'll go over next week. But this is the general gist of what 3A assembly is. It stands for 3 antibiotic assembly, because typically each of these plasmids are going to have different antibiotic resistance on them in order for you to screen them afterwards. But that's a later thing. So I try to make a flow chart of kind of what's happening here. So this is your prefix part. This is your promoter. This is your suffix part. This is your coding sequence for whatever protein you want. And this is the backbone. Luckily, iGEM already sends us pre-cut backbones. In the field, you typically cut the backbone with what sites that you need, but they already cut it for us. So we just have a linearized plasmid backbone with the sticky ends on each end. So we don't have to cut that. It's easier for us. So starting here, the 3A assembly calls for cutting this with E and S. So EcoR1 and SPE1, your prefix part. So you cut it on both ends. The EcoR1 matches to this. This is where you want to insert it, right? So this sticky end matches with this sticky end. And on the suffix part, on this end, you're cutting it with the P. So on the right-hand side, this P is going to match up with this P. So now you have pretty much this part attached to this end right here, and this part attached to this end right here because the S's match up. But in the center, what's happening here? So you have an S and an X. They don't match, right? How are they going to connect? Well, luckily, SPA1 and XPA1 have the ability to attach together and form a fully, ligated part. However, you have to note that once you do this, unlike the other parts where you can cut them afterwards, because technically it's just a duplicated EcoR1 site, this is a mixed site now. So it has both the properties of an SPA1 site and an XPA1 site. The restriction enzymes aren't going to recognize that because it's mixed. And so once you ligate these together, it's a one-way street. You can't cut it. with at least these restriction enzymes. Just to wrap this up, so you have your prefix part. You're cutting it with EcoR1 and SPA1. You have your suffix part. You're cutting it with XPA1 and PST1. The E on this part with the sticky end overhang is going to go to the E on this. The P on this is going to go to the P on this. And the S right there, is going to attach with the X right there on this part to make a fully cylindrical plasmid once you ligate them together, and it forms this. Now it's kind of complicated. I know for like just viewing this from the start, and I'm not the best explainer in the world, but if you look at this and if you search up 3 assembly on Google and you read the page on iGEM, they give a pretty good explanation of what it is and the protocol for doing it. But overall, you don't really have to worry about this. The main thing I want you to take from this lecture / meeting is you cut the prefix with E and S, you cut the suffix with X and P, and you don't have to cut the backbone. Next week, I'm going to be going over 3A assembly in detail, this whole thing in detail. You can worry about it then. So doing the double digest, this whole procedure has more of a research focus. So you have to know what you're doing beforehand before you actually start doing the procedure. Otherwise, you risk the chance of really messing up. So the goal here is to do a few days of research before you start, putting the parts, cutting the parts, and then you finally have all the enzymes that you have determined for each part, and you can begin doing the procedure. So first, you're going to want to pick your parts. So say your promoter and your coding sequence. You're going to pick what 2 enzymes you need for each part. I went over that in the last slide. And next, you're going to want to get the buffers that you're going to need for the protocol and the tubes. So you're going to need the tube for each part with each 2 enzymes. So one tube with one part and 2 enzymes, another tube with 1 part, 2 enzymes. Next, you make sure that the concentrations and volumes follow the guidelines in the lab manual. So you're only going to want to add so much of the enzyme and so much of the DNA and so much of the buffer. The lab manual goes into that detail. Overall, your goal is to get each tube to have 25 microliters of solution in it. That's for if you're doing a half solution. If you're doing a full, it's going to be 50. Typically, you'll always use 1 microliter of each enzyme. And your goal is to get it to 25 by adding water or a buffer, just to make it even. So next, you add the components to the tube. You're going to add the 2 enzymes last, though, because these enzymes are very, very fragile. If they stay in a room temp environment for an extended period of time, they're going to start denaturing. They're very expensive. I'll get into that at the end of this, actually, because we're ordering some enzymes. But keep those on ice as much as possible. Take them out of the ice quickly, pipette it out, put it into the tube, put it back in the ice. After you add all the stuff to each tube with the enzymes, you take each of those tubes and put it in the thermocycler, which is the machine that changes temp based on time, and you put it on the iGEM digest protocol. Austin and I have done this, right? Yeah, so yeah, you put it in the protocol, you wait like an hour and a half. It's a pretty long protocol, and you come back later and it's done. You put it in the freezer. So the parts are then cut with the sticky ends on each. So next week, I'm going to be going over gel electrophoresis. I think I'll touch on 3A assembly as well. Gel electrophoresis, in a nutshell, is just using electricity, electrodes, in order to separate DNA. I don't know if you've done this before. It's pretty commonplace in wet labs. But you have a positive and a negative electrode, and the DNA runs to the red or runs to positive. So it can separate depending on the size of the part.